Welcome all to Neuroscience 1, and today we'll be talking about the membrane potential. This is my first video that actually goes over some important concepts and mathematical notions, so this will be a long video, and this is also one of my first videos actually filming in this format, so please bear with me if I'm just going through some technical problems or, if I'm slow or not. So we'll just get started right away and start by talking a bit about the membrane potential. So what is the membrane potential? Essentially, the membrane potential is the difference. It is the difference of electric potential across the membrane, obviously. So what I'm connecting this to is the electric potential. So the electric potential, if you've taken a physics class before, it's quite similar to what electric potential is there, right? So say this was part of my circuit, and I had a bulb over here, something like this, and it's shining. And if you want to measure the voltage or the difference in potential across this bulb, you would want to put a voltmeter over here and over here to actually measure what is the difference in potential across these areas. So voltage or potential is the difference in potential between 2 points of space, or in this case it would be two liquids in a medium. And basically what the potential does is it describes the strength and direction of electrical forces that would direct the flow of the electrical field. And the electrical field also dictates the strength of the force that will be applied on, say, an ion moving through that gradient. So, connecting this to the membrane, say this is our membrane. It's kind of a lipid bilayer, so I'll just draw it like that instead. We have heads and tails like this. So this is our membrane, and over here, if I can find my cursor, over here we have extracellular fluid. I'm coloring this red because usually red denotes positivity, and positivity not being positive in life. I mean positivity as in there's more positive charge. And obviously to contrast that, I'm going to have a blue color be the intracellular region. So membrane potential is the potential across this thing. So we would put something on this side of the cell and this side of the cell, and then we would connect them together, and using this we can find out what's the potential here and what's the potential here. And to find difference in electrical potential, let's say this is V in and this is V out. So the difference, or also known as the membrane potential, would just be V intracellular minus V extracellular. So, as you might have noticed, we're going to take what the outside is. We're going to take the outside potential to always be our reference point. So this is our reference point, meaning we will consider this to be our zero or our ground. So if I were to say the membrane potential, or if I were to say the intracellular region is 50 millivolts less positive than the outside, I'm essentially saying this is 50 millivolts more negative than this. So this would be 50 and this would be 0. Or this would be 50 and this would be 100. Or sorry, the other way around. This would be 100 and this would be 50. So that way you can see more negative also equates to less positive. So in this case, if you consider this case, say the outside is 100 millivolts and the inside is 50 millivolts, if you were to use this equation over here, you will still get 50 minus 100 is equal to negative 50. Okay? I do realize I'm kind of going off camera. Just realized that. Okay. There. If anything was important was missed here, I'll just kind of do that. So that's shown. And I'll keep in mind that I have to shift. Okay. So now that we've covered like a basic, we have a basic understanding of the membrane potential. We also need to know that the potential will remain constant for any period at which the cell is not propagating any signals. And we'll get into this more when we talk about action potentials. But when an action potential is formed, the cell initiates a signaling response that goes down other neurons and goes down other neurons to transmit a message, whether that be a motor function or a sensory reflex or whatever it might be. But yeah. And another thing that, since we're on this topic of, voltage and how exactly stuff works in an electrical sense, we should also think about the simple properties of the electrostatic force. So the electrostatic force, I'll just write that over there. The electrostatic force. The electrostatic force is the attractions and repulsions of charged particles. So why I'm kind of introducing this notion is because obviously we're not in a physics class, so we're not talking about like particles in space. We're talking about ions that are present in, say, the intracellular fluid or the extracellular matrix. And the electrostatic force between those two becomes really, really important when we start talking about, the potentials and how the Nernst potential works and equilibrium. But I'm just introducing it right now so we can talk about simple ionic attractions. So we know that, or this, hopefully you know this. This will just be a review if you already know this. But a positive particle will be attracted to a negative particle. That's not blue. I tend to color code stuff. So the negative will be a blue color. Hopefully you can see that. And so this is an example of an attraction. So these two particles will attract each other. There will be an attraction force. However, if I had, say, a proton and another proton, that's not going to work. So these two are going to repel each other. And the same thing goes for when we have two electrons together. I realize that's not the same shade of blue, but whatever. So the only attraction force you'll ever get is when you have two different particles, two differently charged particles. And why this is important is because, like I mentioned before, there's a lot of movement that goes from the inside of the cell to the outside of the cell and vice versa. And that's due heavily to the fact that there is a lot of there's a lot of attraction and repulsion going on, but mainly repulsion between, say, potassium and sodium and potassium. So together, these kind of make up something that's known as a sodium-potassium pump, which we'll talk about later. But that's just a functionality of how we could characterize the flow of ions from one side of the membrane to the other side of the membrane.